for the introduction. Thanks to the organizers of this session for the opportunity to present some thoughts and some results, and thank you also, Professor Gao, for your excellent presentation, because it has really provided already the framework for what I'm going to talk about. And you have been concentrating mostly in the Pacific, whereas I will be concentrating more in the Mediterranean Sea and the Atlantic. So hopefully together we will provide a general overview. As you already explained, the reason we are interested in these events of nutrient deposition is basically because the biological pump, the group of organisms in the plankton that contribute to the export of carbon towards the interior of the ocean, can be affected by different inputs of materials coming from the atmosphere. You know, you can have inputs of iron, phosphorus, nitrogen, but also potentially toxic components such as copper, for instance. So we are interested in understanding this connection between land ecosystems and ocean ecosystems through the atmosphere, whereby different nutrients can potentially affect the biological pump. Most of the ocean is limited by nutrients, but this limitation can actually occur in two different ways. You can have high nutrient, low chlorophyll waters in which nitrate and phosphate and also silicate is present in sufficient concentrations, but iron, the micronutrient iron, is not. Therefore, you have a limitation of the system because of the iron. Whereas in the subtropical gyres, typically you have a low nutrient, low chlorophyll concentration system in which macronutrients, typically nitrate, are limiting the system. We know this for a fact because bottle incubation experiments and in situ nutrient addition experiments have indicated to us that, for instance, nitrogen, which is the green circle there, is limiting these subtropical gyres, typically, whereas iron, as I was mentioning, is typically limiting in the HNLC waters. So basically, that's the background to the story. And the way I've organized this overview is I will be trying to show how different taxa can respond differently to atmospheric deposition events, and in particular to dust deposition events. So I want to emphasize that different components of the community can easily show different responses. I will move on to discuss some changes in key metabolic rates such as photosynthesis, nitrogen fixation, but also heterotrophic processes such as respiration. And I will try to convince you that the balance between the responses of the photoautotrophs and the responses of the heterotrophs is key. And finally, I would like to give a more broader picture of the role of dust deposition and atmospheric deposition by looking at large-scale variability in some biogeochemical fluxes that can be ultimately driven by changes in dust deposition or atmospheric deposition. So I would like to begin with a number of experiments that we did in the Atlantic back in 2007 and 2008, in which we added Saharan soil and collected dust to bottles and tried to analyze what the response of the community was in terms of the abundance of different organisms. And just to give you a broad idea, I will not be getting into much detail here, but different groups of organisms, you have here the Synechococcus and Prochlorococcus and the picoeukaryotes, for instance, tend to show different responses. They are not really synchronized in the way they respond to these perturbations. For instance, Prochlorococcus and Synechococcus overall tended to show a negative rather than a positive response to the dust addition. Similarly, in the heterotrophic bacteria, we found that, for instance, the SAR11 group, which is in the upper right-hand corner, tended to show, again, a negative response. So for some reason, these guys here didn't like the dust much, whereas the gamma protobacteria, they tended to show a positive response. This has been also seen by other authors. For instance, Polly Hill, also in the North Atlantic subtropical gyre, found that both Prochlorococcus and the SAR11 bacterial group showed a decrease in their rates of uptake of methionine after they had been exposed to dust perturbation. So again, you can see here a decrease in the uptake rates of both Prochlorococcus and the SAR11 group, reduced metabolic activity, therefore, in the presence of dust. This has been also seen in the Mediterranean Sea. This is Barak Herut's work. He found, basically, that after a heavy dust storm, the abundance of Prochlorococcus decreased dramatically. But on the other hand, bacterial activity, in terms of the uptake of a substrate, increased. So again, we have these taxon-specific responses to dust. Adina Paitan also has shown in the Gulf of Aqaba, in the Red Sea, again, a reduction in the abundance of picoeukaryotes and Synechococcus in response to Saharan aerosol. So we have a negative effect of Saharan dust on the abundance of some organisms. And just finally, to close this picture about the possibility that sometimes these events of dust deposition can actually have a negative effect on the biota, just a more recent example of a correlation analysis conducted by Jordi and co-workers, who found that decreases in chlorophyll concentrations immediately after events of dust deposition that were rich in the element copper. So therefore, what we have is a negative relationship between copper concentration and changes in chlorophyll concentration. How do metabolic rates respond to these dust deposition events or dust perturbations? It has been known for a long time that there is typically, and Professor Gao was showing this as well, that there is typically a positive response, particularly in terms of chlorophyll concentration sometimes, and also in terms of primary production. So here we have two examples with bottles. These are bottle incubation experiments. And in both cases, in the Western and the Eastern Mediterranean, there is clearly a positive correlation between dust addition and an increase in primary production. Of course, bottle experiments can be criticized, but mesocosm experiments, which reproduce much better in situ conditions, have also seen that, have also provided the evidence that primary production can be quite responsive to dust deposition. In this case, this was the Dune mesocosm experiment done by, coordinated by Cécile Gueux near Corsica in the Mediterranean Sea. They found a clear increase in primary production in response to the dust additions. Nitrogen fixation can also be quite responsive to dust additions, and this makes sense because dust carries iron and also phosphorus, and these two elements, iron and phosphorus, can typically be limiting for nitrogen fixation. These are complicated experiments. I would just want to concentrate on one aspect. These D1 and D2 treatments here, they correspond to dust addition experiments. They use Saharan dust, and they found clearly an stimulation of nitrogen fixation. It is not clear to which extent that stimulating effect was due to iron or to phosphorus or both. Again, the Dune experiment in the Mediterranean Sea, they found a positive effect of dust on the nitrogen fixation rates. But now things become a bit more complicated. If we consider bacteria and their activity in terms, for instance, of bacterial production or also in terms of bacterial respiration, there is also an stimulating effect of the dust. So it is quite often the case that the bacteria also respond to the dust addition, and in fact they can respond even in a stronger way than phytoplankton do. So these are various different examples, all in the Mediterranean, of an increase in bacterial activity or bacterial respiration as a result of a dust addition, either in bottles or in mesocosms. Now, why is it important to consider both autotrophic and heterotrophic processes? The reason is that if we have, due to anthropogenic global change, an increase in atmospheric deposition, and if that increase in atmospheric deposition stimulates mostly the primary producers, we have a negative feedback on atmospheric CO2, because basically we are stimulating the biological pump due to these additional nutrients coming in from the atmosphere. On the other hand, if the stronger response is observed in the bacterial production and bacterial respiration, then we have a positive feedback on atmospheric CO2, because if the stronger response of the organisms is that they respire more, then they will be sending back CO2 to the atmosphere. So that's why I think it is important to consider both autotrophic and heterotrophic processes together. And this is what we did coming back to these experiments in the Atlantic. We measured both primary production and bacterial production, and it's a bit of a mess, really. In some experiments there was an stimulation of bacterial production, in some others there wasn't. And with bacterial production the same thing happened. You know, sometimes you had a response, sometimes you didn't, and sometimes even the response was significantly negative. So it was not easy really to understand what was going on. One possibility that I'm going to suggest to you is that the response of the community may be dependent on how oligotrophic the system is. We had 8 experiments here, and they were all oligotrophic waters, roughly speaking, but they were not all equally oligotrophic. Some of these waters were extremely oligotrophic, and some others were only moderately oligotrophic. So basically there are a number of indicators for this. You can compute upward nutrient fluxes, or you can also have a rough indicator such as the depth of the nitrocline or the euphotic depth layer, which can give you an idea of the fact that some systems, some stations in this study were more oligotrophic than others. So we just decided to consider the depth of the euphotic layer depth as a proxy for the degree of oligotrophy. And when we plotted the response of primary production and bacterial production against the depth of the euphotic layer depth, which I insist is just a proxy for a degree of oligotrophy, we found that primary production tends to be more responsive to the dust addition in moderately oligotrophic systems, whereas bacterial production tends to be more responsive, and in fact dominates the response of the biota in more oligotrophic settings. And therefore the hypothesis that we are putting forward is that the degree of oligotrophy, how much oligotrophic an environment is, can actually be determinant to understand the balance between primary production or phototrophic versus heterotrophic responsiveness to dust deposition. We put together all our measurements, and what we found overall is that in general changes in metabolic rates are greater than changes in abundances. Okay, so rates tend to be more responsive than stocks, and that probably suggests that bottom down grazing control prevents huge changes in the abundance of different organisms when dust comes, but metabolic rates do tend to change more. And within the metabolic rates, interestingly enough, it is the bacterial rates, such as bacterial production and community respiration, which is typically dominated by bacteria, which tend to respond more, even more than nitrogen fixation or primary production. This is just a collection of more than 30 in vitro bioassays of dust addition experiments done by Cliff Law and Cécile Gueux. And what they found, again, with this overview, is that, again, the highest responses observed in terms of positive response to the dust take place in bacterial production and bacterial respiration. There is certainly a positive effect on primary production and nitrogen fixation, but it's a bit smaller. And a tendency to sometimes have actually negative effects on the abundance of some organisms such as Prochlorococcus. Although obviously the dispersion between different experiments is large. So I would like now to conclude with a section on broader perspective of the impacts of these dust deposition events. And we are quite familiar with pictures such as this for the Atlantic, in which we can have large, really large events of dust storms that covers hundreds of thousands of square kilometers, basically. So the Atlantic really provides a really nice laboratory to understand the large-scale impacts of dust deposition, because there is a really clear, marked asymmetry. In the North Atlantic, we have large deposition fluxes, whereas in the South Atlantic, we have much smaller deposition fluxes. Therefore, we can try and understand what the impact is on the biology. And this is work that Mark Moore and co-workers have done. What they found is basically that this asymmetry in the deposition fluxes in the Mediterranean, you can see here with aluminium. This is dissolved aluminium, a proxy for anthropogenic, not anthropogenic, material from the desert. Okay, this is a proxy for the desert material deposited onto the surface of the ocean, and these are dissolved iron concentrations. There is clearly a marked asymmetry. This is the north, and this is the south on the left. Okay, as a result of that, what we find is a really different behavior of nitrogen fixation. Nitrogen fixation is much higher in the northern hemisphere than it is in the southern hemisphere, in the central Atlantic Ocean. Because there is more nitrogen fixation, there is an additional consumption of phosphorus. And as a result, phosphorus concentrations in the bottom right panel are clearly lower in the northern hemisphere compared to the southern hemisphere. This has an impact on the balance between these two elements, nitrogen and phosphorus, as potential controllers of primary production. Because in the North Atlantic, nitrogen is the primary limiting nutrient, but phosphorus is also co-limiting. This was beautifully shown here by Matthew Mills and co-workers in their Nature 2004 paper. What they found is that if you add both nitrogen and phosphorus, you have a stronger response than if you only add nitrogen. Okay? These are three different locations in the tropical North Atlantic. So basically, the fact that there is more nitrogen fixation as a result of the enhanced land deposition atmospheric deposition of iron, the fact that there is more nitrogen fixation in the north explains that there is more phosphorus consumption, and the fact that there is less phosphorus available explains that phosphorus can also co-limit primary production. This does not happen in the South Atlantic. In the South Atlantic, only nitrogen is limiting. Even if you are nitrogen in combination with iron or phosphorus, or even if you add the three nutrients together, there is no additional response as if you only add nitrogen. A similar kind of pattern can probably occur in the Pacific Ocean, and this is work by Shiozaki and co-workers. What they found again is that the largest flux of nitrogen fixation does not take place in the more stratified waters, which would be, in the equatorial region. The largest nitrogen fixation they measure coincides with subtropical latitudes where more iron is coming from the Gobi Desert, probably. And again, this leads to a depletion of phosphate. So we can have probably the same mechanism operating in both basins, both in the Atlantic and in the Pacific. Now, I would like to stress the differences between HNLC and LNLC regions in terms of their ability to respond to deposition events. This is a diagram prepared by Cécile Gueux, and what she wanted to illustrate here basically is that, particularly if you consider an enclosed basin such as the Mediterranean Sea, which is also surrounded by heavily populated areas, you have a mixture of aerosols coming in, mixed aerosols. There is natural aerosols combined with anthropogenic aerosols, whereas in remote systems such as most HNLC regions, you have a tendency to have aerosols that are dominated by natural sources. But now, because of differences in the stoichiometry of primary production, and by stoichiometry I mean the proportions in which different nutrients are incorporated into organic matter, because of differences in these ratios that I will be showing in a minute, the ability, the potential for response to one of these events of dust depositions in these two types of regions is quite different. And in fact, in most cases, when you compare the results of in situ addition experiments, in most cases, low nutrient, low chlorophyll concentration regions, in which typically nitrogen, nitrate is the most limited nutrient, have a limited ability to respond to the dust or to the nutrient addition, more generally speaking. So this would be indicated here on the right. In these regions, typically you have a limited response of the photoautotrophs compared to a quite clear response of the bacteria often. But in fact, most of the time there is not a huge change in the abundance of the organisms. And there is no evidence that neither iron nor phosphorus control the production or the export of organic carbon in these systems. Whereas if we consider HNLC regions, the addition of iron typically triggers a huge response, both in terms of biomass, but also in terms of metabolic rates and even in terms of carbon export. Now, the stoichiometry is behind this difference because the carbon to nitrogen ratio and the carbon to phosphorus ratio, it's much lower than the carbon to iron ratio. The carbon to iron ratio can be extremely high, between 10 to the 4 and 10 to the 5. That means that if you add a really small amount of iron, provided that other nutrients such as nitrogen and phosphorus and silica are available, the potential for the community to respond is quite large. And these are just an example of these, some calculations that I've done, assuming measured contents of nitrogen and iron in dust, and assuming measured parameters for solubility and bioavailability. And basically the idea here is that a typical dust deposition event can provide perhaps around 0.2 micromolars of nitrate, and that can translate into an increase of chlorophyll of maybe 0.1 or 0.2, which can be quite limited. I'm talking here about a nutrient limited, low nutrient, really low nutrient, low chlorophyll concentration, rather than a coastal sea where obviously nutrient availability is probably higher. Whereas what happens in an HNLC region, if you have an increase of iron of maybe only 2 nanomolars, which is really small amount, but if you consider the carbon to iron ratio is so high, then that increase of iron of around 2 nanomolars can translate into what we call a bloom, really, because it would be an increase of a chlorophyll concentration of around 2 micrograms per liter. And I would like just to move on to consider a different way in which materials can fertilize the surface ocean. These are not dust deposition events coming from deserts, but volcanic activity. Volcanic activity is known to be a source of iron, and that iron can stimulate primary production and the abundance of phytoplankton. Just some examples of these really impressive events, although obviously very sporadic and not frequent at all. But this was done by Roberta Hame and a few years back, and what they found is that because of the eruption of the Kasatochi volcano in the Aleutian Islands, there was a bloom of phytoplankton that year that has not been observed in previous years. That was August 2008, and in fact they just happened to have a time series of measurements in that region. So the dark dots here indicate previous years, whereas the blue and the red dots indicate measurements collected during August 2008, just immediately after the eruption of the volcano. And you can see that there was clearly a stimulation of chlorophyll concentration and also photosynthesis. And a more recent example, maybe many of you remember the eruption of this volcano, Icelandic volcano, Eyjafjallajökull, or whatever you pronounce it, back in 2010. Basically, that was like April, May 2010. There were considerable disruptions to air traffic throughout Europe back then. And, well, Eric Achterberg and colleagues just happened to be at the right time in the right place during that eruption, and it was obviously a number of cruises that was already scheduled and to occur, and they were just there. And in fact they did 3, not 1 but 3 cruises that year. So they were able to measure iron concentration, and you can see that underneath the plume, the dust, the ash plume from the volcano, you have a clear increase in iron concentration compared to regions that were not affected by the ash plume. And during the eruption there was an increase in chlorophyll concentration compared with the background climatological values. And again, if you look at the nitrate concentration, there was an additional consumption of nitrates that year. This is a relatively little known HNLC region. This region, the Icelandic Basin, is a region in which you can easily have relatively large amounts of nitrate and phosphate after the spring bloom that are not depleted. And the reason is, as Mark Moore and others have shown, that there is iron limitation. So it's an example of a seasonal HNLC condition. And well, I will not be showing this to you, but they also did bottle addition experiments, and they were able to prove that the volcanic ash had this stimulation effect on the biota. And just to conclude, I would like to consider a little bit export, because at the end of the day, biological pump is about transporting carbon into the interior of the ocean. So we want to know to which extent there is a linkage between atmospheric deposition and vertical fluxes. This is a beautiful database, more than 4 years of data in the Mediterranean, in which they have collected dust deposition rates together with vertical carbon fluxes. The location of the 2 stations is slightly different, because the DIFAMED is a bit further from where the dust collector is in Villefranche-sur-Mer, but also they also have another atmospheric input collector in Corsica. Anyway, what they found is a really strong correlation between dust deposition and vertical fluxes. So you have basically that when there is a strong event of dust deposition onto the surface of the ocean, vertical fluxes of material tend to increase. But interestingly, it is most of the time, it's not that the biota is fertilized. What happens is that the mineral particles coming in actually trigger the export of organic matter that was already present in the system. So it's not necessarily a fertilization effect. It's more an effect of an enhancement of vertical velocities due to aggregation induced by the input of mineral particles, together also with the effect of grazing of mineral particles and the compactation and the increase in the density of the fecal pellet, so what we call the ballast effect. So there's, as I was mentioning before, this really strong correlation between the flux, the vertical flux of particulate organic carbon, and what they call the marine lithogenic flux, which is basically minerals coming in from the Sahara Desert mostly. So again, I stress this point, the highest particulate organic carbon fluxes were measured, were linked to these lithogenic events through this grazing ballast effect. Just to mention an example of the dynamics in the Pacific Ocean, again, this is an example of a correlation between dust events and lithogenic fluxes. That was measured at station Knot on the North Pacific, and also at station 50 North there has been a correlation observed between dust events and the vertical flux of opal. Obviously, if we want to have an integrated perspective, we don't require modeling, because it is the only way to have a high resolution picture image of what the patterns may be. And this is work by Krishnamurthy and co-workers. And what she found here is that in general, the amount of material coming in from the atmosphere is relatively small compared to the amount of material that is exported. This gives you an idea of, and Bea was asking this question before, what's the contribution of these atmospheric deposition pathways to the input of new nutrients to the system. Because at the end of the day, assuming steady state, the sinking flux of silica or organic nitrogen, iron, phosphorus must be compensated by an input of new material. Okay, so what they come up with is these global estimates for how much the atmospheric flux contributes to this export flux of phosphorus, iron and nitrogen to the interior of the ocean. And it's 5% globally for nitrogen, and it's less than 0.5% for silica and phosphorus. But for iron, it can easily be around half. That means that around half of all the iron that is exported towards the interior of the ocean actually comes from the atmospheric pathway. And I will leave you with my conclusions there, and thank you for your attention, and we'll be happy to take any questions.